Let me see though. Oh, I guess here, glued. This is an interesting bit of the thorax. So you see the little spikies here. While it can't be proven, it is hypothesized that these little spiky bits are to prevent like birds from predating on them. And again, I say it can't be proven because to me, the definitive proof will be to get ant that doesn't have this. If you can genetically make one without a spikes and then put them in the wild and then have the ones with spikes and see which ones get eaten and which ones don't. Instead, what they've done is like they'll take Play-Doh and put the Play-Doh like with some spikes on it and just see what happens. And like glued, I don't really know what to make of that, right? Like if it's something that's legitimately finding like Play-Doh and spikes, but is it really that's yeah, so that's the thing, right? Is it's not a natural substrate that they go for. Yeah, exactly, Sparky, the odor. And Sparky, this is something that they've been using for other experiments too, and not just in this one, right? The structural color, where they're trying to identify what structures and colors are like why they evolved, and is it maybe to keep away predators? How would you approach imaging those hard to not destroy nerve connections? Yeah. So Demon, you can embed it in resin and then just take really, really thin slices through the resin. You can do something like that. You can also remove the exoskeleton's color via a protocol for with hydrogen peroxide, and you basically melt away the outside. And that is a way to get the image. You can then put the whole 3D sample into the microscope too, and you can see what's inside. Those are labor intensive and pricier ways of going about it. So it just depends on how important it is to your end goal. So here is the next head. You can see the eyes quite nicely here. You can even see some of those like thinner regions that you can kind of see through with the lighting. It's like they're a little bit less dense. It's probably more just because the muscle is kind of collapsed inward just over time. But yeah, that's a neat little soldier ant head for us. Let's go ahead and I want to just see if there was an injury on the outside of this skull too. Doesn't look it. Here is where the neck goes. Y'all see this bubble right here? That is where the neck connects. So werewolf, I and nod. The only way to release these mandibles is to crack open the top of the skull and snip here that connects here and here that connects here. You cut those and the mandibles will release by the wayside a little bit. So like that's why this is a little bit more thinned out, but all of these darker regions are much like connected in that muscular tissue. That's of how it's preserved. But yeah, werewolf. I mean, I could try, but it's probably just gonna snap the mandible off instead of actually being open. The only way that I've seen is you cut a little window here and cut a little window here as well, and those two are what gets you like views on the inside. Head spikes more on this one too, so you can kind of see it werewolf here and here, but it's still curved like. So this head isn't level. This head right now is like this. So the mandibles are facing downward and the top of it is floating up. So we get to see a little bit more around on the backside of the head. All right, this is a leafcutter ant, Shark Geek. These are leafcutter ant soldiers. And we're popping over their heads. They take a look inside. Let's see. I'm going to try glued to get another image like we got last time with the brain still in place. It is going to go a little blurry right now for y'all. It's unfortunate that I can't get the same field of vision like that y'all get, but it's just two different settings, unfortunately. This specimen, y'all still had the abdomen in place. We'll see what's inside the abdomen. We should find shriveled ovaries. There's a little window inside the skull, and the brain right there. Actually do a little bit more of a zoom. So we've pulled back the layer of exoskeleton, and this structure right here, that is the brain. So it's behind this almost Y-shaped bit of black tissue. That is the size of the main part of the brain. And then you can kind of see there's a strand going down this way, connecting to this eye. And there's another downward facing strand connecting to this eye. So this, again, like relative to the rest of the head, that's the brain tissue. The rest is all muscle tissue. And you can see how little of the brain is actually kept of brain tissue, or the head is kept of brain tissue. The rest of it is mainly just muscle tissue. Pretty cool, right, y'all? That's one of my favorite views to get something like this. Actually, let me show you. I have an image. We'll cut back to this one because I do want to pop out the brain But this view, like a larger zoom, we can do a little bit more zoom. Yeah, I just want to show you all this particular stain. So I've done this dissection before with a different kind of imaging. It's called light sheet microscopy. Oh, purple cat, the colors are real. There's not artifacts. This is the color that you're seeing here is the real color. Some are a little bit brighter because of the exposure, but their color here is totally the same thing. This is not color changed at all. But yeah, here, these are looking at marking the muscles. And so if we did the same stain, so this one pumpkin, this like red is something that we added in. So when it glows, it just glows a different kind of color, but we pseudo colored it. And then it's black and white here because this microscope can't image color real specimens. This can image color real. Why do we choose red contrast? I think actually it may have been a red stain as well. You can change it to whatever color you want, Sparky. Like even if the microscope image picks up red, you can change that to be whatever you wanted. Let me see if we can get an image. Okay, you can kind of see it. Do y'all see this right here? So remember, I've taken the back of the head out, right? So light is shining now up through the head. This right here is the brain. And so you can see it now because of that contrast that we've got. So this little dark region here. No, the small circular thing, this here ghost, is what is called the ocelli. That is the UV detecting organ on top of the head. That is then connected to the brain here, which is this structure. That's the brain. Circular shadows, this one, this one, this one, this one, those are bubbles on the top and inside. So there's also bubbles inside the head and outside right now. That's just a function of us dissecting. And then all the hairs too. All the hairs will be collecting all the fuzzy bits, yeah. So this, I know glued you'll see, but that strand there and then this strand here, that's the optic lobe. Also, what's neat is we actually got the brainstem here. So this tissue right here is the brainstem. You can see there is one of the connection points here. So there is the optic lobe, there's the other optic lobe, there's the top of the brain, and then the brainstem is here. Usually that breaks when we're pulling out the specimen, but we actually got it out this time. Y'all want to look and see the guts? You want to see what's in the abdomen since we have an abdomen sample? I figure my party people will want to see. So the brain, right, there's no spine, so glued, imagine this bit of tissue here going through the neck, and then it's just like more neurons are stemming out, and then that neuron goes to muscle, and that neuron goes to the ovaries, and that neuron goes to whatnot. So it's just there's no spinal column. It's just more neurons that'll then connect elsewhere in the body. So almost like if you took away our spinal cord and then you just have like a deep spot, like a set of bundle of neurons going into your body and then connecting elsewhere. Think of it like that. We're about to dissect the abdomen of this leafcutter ant, seeing what's inside. Sparky Pugwash was wondering if an ant finds one of the colony ants to seize will move the body. It will, Sparky. Sparky, there is a chemical compound that is released called oleic acid from a dead insect, particularly dead ants. And after it releases the oleic acid, that's the chemical smell that tells other ants that are alive that this one's dead and needs to be taken out. You can even buy oleic acid and use a tiny paintbrush to brush that odorant onto a living ant. And living ants will pick up the other living ant and get rid of her because they'll be like, oh, this ant is dead. And it'd be like, no, it's not. It's still living, but it smells like death. And so they get rid of it. We're just about to pop open this ant's abdomen. We did brain dissections looking into the head of the creature. There's actually the brain right there. There's a bunch of muscle tissue. And now we're going to look inside the abdomen to see if we can find anything delicious. Cricket really wanted us to find the ovaries. Will we find the ovaries? I don't know, but we're going to take a try. So all of the ants will have ovaries, but some of them, everyone but the queen is going to have really, really shriveled down ovaries because they are only turned on if the queen dies as a last-ditch effort to save the genetics of the colony. So there might be something in here that we find, but it's going to be really hard, and the chances of us finding it are going to be quite minimal, but we can still try and take a look and see what we can find. Maybe we'll find it, maybe we won't. Like, it better not be angel. There's the abdomen. You can see it's very fuzzy. Now to pop it open. Just to show y'all a quick look, that white is fat tissue. So that's fat body cells. So we're seeing that the ant at least was fed before it passed. There. Y'all, that popped straight out. There's basically all of the internal organs it pops right out, which is very nice. Makes it easier for us. So there's fat tissue in here. We also have a bunch of these tubes, which is actually quite neat for us to be able to grab. So those are all, they were connected to the exoskeleton. That's what's bringing in oxygen into the body. So remember how we've talked about that they breathe through diffusion? So those are the tubes that are connected to the exoskeleton, and the oxygen comes in and it diffuses into the bloodstream through these tubes. We also kind of see up here at the top, those are some fat cells right up here. And then if there are ovaries, they're buried somewhere in here. I'm going to try to tease apart what's in here. There is no guarantee, y'all, that we're going to find really the pieces in here. Primarily because, again, they're probably shriveled, and you saw how easily it all popped out. If it's popping out that easily, I'm like, ooh, there might be some breakdown product that we're not yet accounting for. But we'll try. We'll try to see what's in there. They're all kind of tubes, Cliff. These are tubes for breathing, but like Sam said, we also have tubes that, for the oxygen transfer. We've got tubes for a bunch of different things in there as well. Valve structure to keep water from getting in those tubes that I'm aware of. I think the hairs also give it surface tension, and that surface tension prevents there from being the possibility of water to be flooding, I think would probably be the takeaway. I don't think that there's actual physical barriers for it. Because one thing that will happen, Glued, if you paint the ant or dip it in paint where the surface tension actually covers it, the surface tension is not sufficient to cover the insects, from being surrounded with the paint, they'll actually choke and drown in the paint. So my assumption there is, it couldn't just close off. It has to be a permanent opening, and maybe the hairs are providing that surface tension. Transport oxygen. They do have blood. It's called the hemolymph. And the way that they transport oxygen is again diffusion. So these tubes go into the body from high concentration to low concentration of oxygen. It goes into the bloodstream and it's collected by the hemolymph and moved throughout. There's not a part per se that where the blood goes in and out from. Instead, there's just a muscle that does contract, and that contraction causes the liquid to pulse throughout the body. And that's more what is giving the opportunity for it to have some pulsing movement of blood cells. Again, working in a different manner, but hopefully something that resembles, it's similar in terms of concept, right? Of how it does work. Let's dig inside this specimen and see if we can find something cool in here. We might not be able to. Just a fair warning, I will try, but cells, tubules. Yeah, I'm not reliably seeing it through the whole surface. Yeah, Cliff, exactly. And then you see more tubules depending on the size of the animal. So the reason that we're able to see this is because the soldier is so big. I've never seen it when I popped it out of the nurse. Granted, I was not looking for it when I did those dissections, but it wasn't just an easy happenstance thing that I found with the nurses, because they're just packed full of a bunch of other stuff too. And here are some fat cells. So this is the fat body tissue. Decomposed. It hasn't decomposed, Sparky, because it's been preserved in alcohol. So that keeps the decomposition from happening. But sometimes the tissue, instead of decomposing, it just kind of falls apart. So it's not as sturdy over time, and it's been in the alcohol, I don't know, for a few years now. So just some of the connective tissue will just kind of break apart because it's in preservative, and so it just makes it very brittle. And if it's a really small piece of tissue like the ovaries that are already brittle in these animals, because they're not supposed to make eggs, right? For this stage, this cast isn't supposed to be an egg producing cast. They're just supposed to be making, taking care of the colony in terms of defense. The only time that these animals make embryos is at the time where the queen is dead and they need to lay an unfertilized embryo, in which case this would be a male. And so they lay an unfertilized embryo, male ant, laying it, the male hatches, and they're hoping that it'll find a female to pass on the genetic information of these animals. Now, whether or not it's going to, that's a big catch, because it's, if it's the wrong season, right, then they won't be able to have anything be able to lay. Even if the male flies out, it's there's no female around. And so it just depends on a lot of things going right in a situation that has gone horribly wrong for that ovary to do anything. And supposedly, Sparky, it's also present in the other casts. It's just more likely to happen in the bigger casts that have more tissue because they have more fat cells that can feed the ovaries versus like the nurses, their bodies also just don't support the embryo of a new male or queen because their bodies are just so small. There's physical limitations there. But yeah, y'all, those are our ant brain dissections for today. We dissected leafcutter soldier ant brains live on twitch.tv, which I think is super cool.